Okay, in this video we are going to be talking about single carbon metabolism. We've looked at many reactions so far, and many of those have required the movement of two or more carbons. But there are occasions when we need to move single carbons in individual reactions. So let's look at the mechanism of what is happening here. The first thing we have to do is start out with this frightening molecule right here. This is called folate. I think many of you have probably heard of folate. And folate, we cannot synthesize this, but the name comes from the word foliage. And foliage comes from your leafy green vegetables. So eat your leafy greens, just like your mother told you, and you will have plenty of folate in your diet. Many individuals do not have this, and so they have to supplement their diet. So folate comes in this form, and you'll notice that there is what's called a pteridine ring structure. The pteridine ring is on the left-hand side, and then you'll notice that this structure here is called PABA, for para-amino benzoic acid. So here's your para-amino in the para position off of the acid, the carboxylic acid, and here's your benzene ring. And some of you probably have heard of PABA. That is what is included in sunscreens. So folate itself is not, we are not able to make it, but bacteria can, plants can. And in order to take the folate up in our diet, we have to have some sort of microbial action, and that is going to add this structure right here, which I think you'll recognize as the amino acid glutamate. Glutamate is going to be added onto the PABA ring on here, and there's a variable number, so that's why I have these parentheses, these brackets around it. And then I'm going to put the letter N here because the number of glutamate residues that are added can be variable. Okay, so that's about all we're going to talk about that end of the molecule. The one carbon, single carbon metabolism, the business end is on the left-hand side between the pteridine ring and the PABA structure. The folate needs to be transformed into something that we really need, and this is going to be transformed by an enzyme called tetrahydrofolate reductase. Tetrahydrofolate reductase is inside our cells, and we are able to take the folate and undergo two successive redox reactions in order to reduce these nitrogens here, this carbon. So we are going to put a hydrogen here, and we are going to put a hydrogen here in two successive reactions. This is not going to use NADH, but rather this is going to use 2-NADPH. So we're going to use our biosynthetic reducing power for this reaction. Now, the numbering on this is critical because we are going to be focusing ourselves on two particular nitrogens. We are going to focus on this nitrogen, which is considered atom number 5 in the ring, and we are going to focus on this nitrogen, which is considered atom number 10 in the folate structure. Now, once we undergo these redox reactions with NADPH and the enzyme tetrahydrofolate reductase, this structure now becomes tetrahydrofolate. And it's very often abbreviated as FH4. And the FH4 indicates folate that has been fully reduced by putting hydrogens on the nitrogen, the adjoining carbon, this nitrogen, and then this carbon. So the molecule is fully reduced. Once we form tetrahydrofolate, this now becomes our active molecule that is going to be used in the 1-carbon transfers. Now, once we have our fully reduced folate into tetrahydrofolate, we can now start performing the preparatory reactions in order to make the reactive substrates that will be the carbon donors in the 1-carbon metabolism. So I'm drawing here just the top part of the pteridine ring and the nitrogen of the PABA structure, just to indicate the N5 and N10. These are going to be our reactive atoms here. So the next step we need to do is we need to donate a carbon onto this structure in order to form the substrate that's going to mediate the 1-carbon reactions. So the donor of the carbons is going to be the amino acid serine. Serine, remember, has the CH2OH on its side group, and that group is going to be, carbon is going to be the donor, and the resulting substrate is to form the amino acid glycine. So remember, serine to glycine. So remember, glycine has the hydrogen as the side chain. So it's this carbon right here that will be donated into forming the substrate for the 1-carbon reaction. Now, the structure that's going to form is going to be the N, which is N5, CH2. Here is the N10. This goes off into the benzoic ring. And then the single carbon that's coming off of the serine is going to be bonded to these two nitrogens. This is forming a methylene structure. And the name of this structure is N5, N10, methylene tetrahydrofolate. So this carbon source here, bonded to these nitrogens, is a good leaving group. So this is going to be an excellent source of carbon as a donor for the next reaction that is necessary. Now, the other reaction that can happen is that this N5, N10 methylene tetrahydrofolate can undergo an oxidation reaction, and this oxidation reaction can yield this structure. This structure here has been oxidized from the CH2 group here. And notice it is now on the N10 atom. So the name of this is a formal group, so the name of this structure is N10 formal tetrahydrofolate. Now, once again, with this double bonded oxygen here, and you've got an electronegative group here, this nitrogen, this is also a good leaving group. So the carbon source for 1-carbon metabolism from either the methylene structure and the N5N10 methylene tetrahydrofolate, as well as the formal structure on the N10 formal tetrahydrofolate, are both good carbon sources for 1-carbon transfers. They're both good leaving groups for these reactions, and the subsequent enzymes that will be taking these carbon sources can extract those from the tetrahydrofolate. Now, when that happens, what is going to happen is we are going to regenerate our tetrahydrofolate. So just removal of this carbon will reactivate the tetrahydrofolate structure, restore it, and so we can do subsequent rounds of serine carbon addition and keep the cycle going for when carbon transfers. Now, one problem that we have is that the methylene tetrahydrofolate group can undergo an additional redox reaction, but this is a reduction reaction. And this reduction reaction is going to be using NADPH. And what we are going to form is this structure. So the reduction of the methylene form is going to create a methyl group on the number 5 nitrogen. So the name of this structure is N5-methyl tetrahydrofolate. Now, this presents a problem for us because this methyl group right here is not a good leaving group. In order to remove that methyl group from the nitrogen, it takes a lot more energy than the simple one carbon transfer straight away can handle. So we are going to have to deal with this in another way. This can be used as a carbon donor, but there's going to be a succession of a couple more reactions that we have to consider. Okay, so before we take a look at the complexity of the N5-methyl tetrahydrofolate structure and how we have to deal with that, let's look at how it participates in a series of reactions to make that single carbon usable in the one carbon transfers that we're going to see. So, in order to start this reaction, we need to start out with the amino acid methionine. This is methionine. So methionine, we need to form the proper substrate in order to perform these one carbon transfers in this pathway. And what we're going to do is we're going to take advantage of this sulfur group right here on methionine. And what we are going to form is this structure. What we've done essentially here is that we have added, using ATP hydrolysis, we have cleaved off the pyrophosphate plus inorganic phosphate, and we have put a nucleoside onto the methionine sulfur group in the form of adenine. So this is going to be added to the ribose structure of adenine. But most important, we're adding this large adenine structure on here, and look what's happening to this sulfur group. Sulfur now has 3 bonds, and it doesn't like that. So this will have a permanent positive charge, and this becomes an unstable sulfonium ion. The name of this structure is S for the sulfur, adenosyl methionine. It's abbreviated SAM. This structure here, SAM, S-adenosyl methionine, is one of the major substrates involved in one-carbon transfers because of this unstable sulfonium ion. Here is the carbon that will be used in these transfers. Now, many enzymes will use this SAM as a cofactor, and so we will have a reaction where a single carbon is needed, and we can call whatever molecule that is as the acceptor, and we can name the product the methylated acceptor. And what we end up having left is this structure. This structure, notice, has the adenosyl group remaining on the sulfur, but the methyl group is gone. And that methyl group is the carbon donor that has been added to the methyl acceptor. This structure here is called S-adenosyl homocysteine. Now, as you know, these reactions are not consuming these products because these have to be restored so that we can continue these single carbon transfers. So the next step in the reaction is to release the adenosine nucleoside, and what we result in is having that sulfur free for the next step of the reaction. This structure here is called homocysteine. And all we've done was taken the adenosine off. Okay, now, in order to restore the methionine, to continue the formation of SAM, S-adenosyl methionine, we are going to convert homocysteine into methionine. And the very simple way to do that is, let's go back to the whole reason we started this, and we are going to use the N5-methyl tetrahydrofolate. And this is where we are able to extract that methyl carbon to restore the tetrahydrofolate structure and take that methyl group on the N5, add it to the homocysteine, to that sulfur, and then we generate methionine to complete the cycle and continue for the next round. Okay, so now we've seen the formation of S-adenosyl methionine using the N5-methyl tetrahydrofolate as a cofactor, but recall that I said that there was a problem with donating that methyl group directly to the reaction. And so what we saw was that homocysteine is forming methionine by using this N5-methyl tetrahydrofolate. Okay, so the problem with this is that the N5-methyl, that methyl group, is not donated directly to the homocysteine group. we have to go through another intermediate. And that intermediate is adding this group, that methyl group, on vitamin B12 in order to form the vitamin B12 methyl complex. In doing this, what we have generated is tetrahydrofolate, which can now undergo additional rounds of serine addition of that donor carbon and form the N5, N10 methylene tetrahydrofolate. And then recall, this can oxidize into N10 formal tetrahydrofolate, or it can reduce back into N5 methyl. We need to go through a vitamin B12 methyl group. And so the vitamin B12 is now going to contribute the methyl group and regenerate B12. Now, a little bit of medical interest here is that the concentration of homocysteine is of interest to cardiologists especially because high levels of homocysteine are associated with cardiovascular damage. And this is primarily arterial damage. The actual mechanism of this association is still being worked out, still not completely understood, but it turns out that it is a reliable biomarker for an individual with associated cardiovascular risk and damage. Now, what happens? How is this level going up? And it's thought to be primarily because of a vitamin B12 deficiency. So this associated damage is thought to occur primarily because we cannot do one carbon transfers from SAM because SAM is deficient. We have sufficient N5 methyl tetrahydrofolate available because of this reduction reaction. However, with the absence of B12, we have a buildup of this, which is going to end up resulting in a loss of sufficient amounts of tetrahydrofolate because this reaction to reduction is preferred over oxidation. And the only way to regenerate tetrahydrofolate is to go through the B12 reaction, and then the B12 becomes the methyl donor to form SAM. A deficiency in vitamin B12 now causes a problem that methyl group on the N5 methyl tetrahydrofolate is no longer able to be released and used, and so this results into a folate deficiency, tetrahydrofolate deficiency. So the way this is typically cured is that if an individual has high levels of homocysteine levels in their blood, that typically indicates that homocysteine is getting backed up because we can't form methionine. So we usually give a vitamin B12 supplement. Now we'll talk in a second about dietary supplements. Dietary supplements of vitamin B12 are sometimes difficult to get inside the body, so usually if a person is in severe B12 deficiency, they'll usually inject it directly into the body. Now, that being said, remember that the B12, sometimes physicians will stop with just going with the B12. But look at what's happening here. This reaction in a B12 deficiency is stopped. So we're building up our concentration of the methyl form, which is going to be depleting the tetrahydrofolate. So this also can lead to a folate deficiency. So knowing your biochemistry, if you ever have this problem where you have high homocysteine levels, which can be associated with indications of cardiovascular damage, and somebody recommends to you taking a B12 supplement, they're absolutely correct. But also, it's recommended now, physicians usually follow the course of also giving a folate supplement also. Okay, so now let's talk about the dietary sources of vitamin B12. Because I mentioned that there's a problem with B12, that sometimes it's difficult to get into the body. Vitamin B12 is a very large and complex molecule, very complex molecular structure, and it also has a sufficient amount of hydrophobicity that when B12 is taken orally, it tends to stick very well, binds very well to proteins. Well enough that the proteins need to be digested in the stomach. Now the combination of low pH in the stomach plus proteases in the stomach causes the proteins to be digested and the B12 is released. Now, there's also cells in the stomach that produce what's called intrinsic factor, another protein that now is able to bind vitamin B12 because it's free from the dietary proteins and form a vitamin B12 intrinsic factor complex. This vitamin B12 complex now enters the intestine and is recognized by cells in the distal intestine, and it's taken up and transferred to the blood. But because it has such a high hydrophobicity, we need to have an associated transporter with it. And so the B12 inside the intestinal cells will release the intrinsic factor, which was its carrier from the stomach down through the intestine, and it will now bind to another protein called transcobalamin. And this will form in the blood a vitamin B12 transcobalamin complex. Now the B12 is safe from binding other proteins. It's got its carrier protein, and then this B12 can go to all tissues that require B12 in order to generate the S-adenosylmethionine. Now, what I didn't mention before, and I'll just mention briefly here, are some of the reactions that are required for these one-carbon transfers. Many of these happen to be in nucleotide synthesis. There are many steps, a number of steps that require nucleotide, during nucleotide synthesis, these one-carbon transfers, and what are we going to use here is typically SAM. The other one that you have seen is the formation of phosphatidyl choline from the precursor phosphatidyl ethanolamine. Remember that ethanolamine, this amine, is a primary amine, and this choline has now 3 methyl groups on the end of it to form this glycerophospholipid. Each one of these methyl groups comes from an individual SAM addition. Other molecules that are required on this is another hormonal molecule that you've heard, epinephrine. So there are a number of reactions. We're not going to go over all of these reactions because they're very diverse, but just be aware of some of these major reactions that require these single carbon transfers.